I forgot. Okay, you're good.
Ready? Okay.
Yep. Thank you.
All right. I think we coming in. Go ahead. I'm going to just start with the introduction. So I thank you everybody for joining us for our CGIPD seminar series. I'm incredibly pleased to introduce Dr. Siyan Cao, who is currently an assistant professor at Washington University in St. Louis on the tenure track, an outstanding physician scientist. I met Siyan at DDW earlier this year when he was also awarded a Inflammatory Bowel Disease Genetics Consortium's R01 to use multi-omics to sort of help us characterize IBD better, which is along the lines of stuff that I do here at UNC with various collaborators. So immediately I wanted him to come and talk about his research here. Siyan received his PhD at the University of Michigan Ann Arbor and then went to med school at Columbia in New York, followed by his residency in internal medicine at UCSF, and then finally fellowship at WashU in St. Louis, where he's now, as I stated, faculty and has an independent lab. He is a recipient of the CCF Career Development Award, an American Gastroenterological Associate Fellowship to Faculty Transition Award, amongst other things. And I'm really excited to hear him talk about his research. So welcome, Siyan, and the floor is all yours.
Well, yeah, thank you so much for having me, and thank you for the introduction. Can you guys hear me well?
Yes.
Okay, great, awesome. Yeah, it's a great pleasure to be here and to talk about some of our recent studies related to inflammatory bowel disease. So yeah, I have no financial disclosure, and yeah, I'm just gonna give like a very brief introduction of inflammatory bowel disease. We know it's an inflammatory condition involving the GI tract and beyond, including two major subtypes, including Crohn's disease and ulcerative colitis. And it's a challenging disease, and the etiology of this condition is still being unclear. And the studies in the past 20 years basically identified multiple pathways that involve the pathogenesis of IBD, and including the innate immune responses, as well as organelle called endoplasmic reticulum, or ER, which is present in most mammalian cells. And my research has been specifically focused on the interaction between the innate immune signaling pathways as well as the endoplasmic reticulum biology. So this is the outline of my talk today. I will first give you a very brief introduction of what is endoplastic or ER stress. And I will be talking about our study focusing on the ER stress pathway called IL-1 alpha XBP1 in two groups of innate lymphoid cells called ILC3 and ILC2. And then I will talk about our recent studies on ER stress in IBD-related complication called Crohn's-like disease of the pouch by single-cell analysis. And finally, I will talk a little bit about our most recent data on perianal fistulizing Crohn's disease. So what is ER stress? Since we know that ER is an important organelle that's for the maturation and folding of proteins in the ER lumen. And when the homeostasis of the ER is disrupted, and there's a condition, basically we call it ER stress. And the ER stress leads to the activation of three protein sensors on the ER membrane. They are called ATF6, PERK, and IL-1 alpha. And the IL-1 alpha is the most conserved ER stress sensor. And upon ER stress, it leads to the activation called XBP1 by alternative splicing of the mRNA. And XBP1 basically can function as a potent transcription factor for pathways such as ER chaperone induction, ER mediated protein translation, as well as inflammatory response. And besides activating XBP1, IL-1 alpha can also mediate the degradation of a number of mRNAs on the ER membrane, as well as interact with pathways such as NF, HBB, and JNK in the cell, and contribute to inflammatory response. And part of my study basically specifically focused on this type of innate immune cells called the group 3 innate lymphoid cells, or ILC3s. And ILC3s basically were identified about 15 years ago by a group of scientists, both in the mice and in humans. And people found that this cell type can control the intestinal homeostasis in a number of disease models. And in the GI tract, the ILC3s are a major innate immune cell subtypes, and it can be activated by a number of environmental factors, including the cytokine IL-23 and the neuropeptide called VIP. And upon activation, ILC3s produce a number of cytokines, including IL-22 and IL-17, and they are important for antimicrobial response as well as epithelial barrier functions, GI tract, and therefore important for the homeostasis of the mucosa. And in IBD patients, people found that the number of ILC3s actually are reduced in the mucosa, although the clinical impact of this cell type in IBD remain unknown. So in this study, I specifically focused on the ER stress regulator IL-1 alpha. And one of the reasons is IL-1 alpha is highly expressed, as you can see here on this panel, highly expressed in ILCs, although the function of IL-1 alpha has not been studied in any of the ILC subtypes. So in this particular study, I tried to address three questions. First is how inflammation affects IL-1 alpha and XPP1 pathway in ILC3s, and how is this pathway regulate ILC3 in GI infection and colitis, and how ILC3 response may become biomarkers or therapeutic targets in IBD. So to answer the first question, I first sort of purified ILC3s from the small bowel of wild-type mice, and I treated the cells with IL-23 and IL-1 beta as combination two cytokines, as well as tunicamycin, which is commonly used ER stress inducer, as well as IL-1 inhibitors. And here you can see the combination of two cytokines is sufficient to upregulate XBP1 by both flow cytometry and qPCR here in the lower panel. And in contrast, other ER stress components such as CHOP, BIP, and GRP94 basically was not affected by the inflammatory cytokines, which suggesting that the activation of this pathway is specific for inflammation in ILC3s. And similar to the ex vivo cell culture, I also look at the XBP1 expression in cloning ILC3s from mice with DSS-induced colitis. This is like a chemical-induced colitis model. As you can see here, there is an increased frequency of XBP1 positive ILC3s in the mice with colitis. And similarly, in the mucosal ILC3s from patients with Crohn's disease or ulcerative colitis, you can see this upregulation of XBP1 in the ILC3s from the mucosa. And we also reanalyze this UC single cell database, which was published a couple of years ago. And here we look at the expression of IL-1 alpha, XBP1, and DNA JB9, which is a downstream target of XBP1. And you can see on this panel from both the inflamed and non-inflamed UC tissue, this is the increased expression of IL-1 alpha, suggesting the activation of IL-1 alpha and XBP1 in ILC3 is present in both mouse and human patients with IBD. And next, I ask, what's the downstream mechanism of this activation of this pathway? Since previous studies already show that the mitochondrial reactive oxygen species, or mitochondrial ROS, is important for the activation of ILC3s, I treated the cells with inflammatory cytokines, and co-treated them with a mitotempo, which is a mitochondrial targeted antioxidant, as well as rotentin and antimycin A, which are electron transport chain inhibitors. Here you can see that the co-treatment with mitotempo or RA are sufficient to downregulate XBP1 in ILC3s, both by flow cytometry and qPCR, suggesting that the multicultural RAS is required for the activation of XBP1, even XBP1 ILC3s. I also treated the cultural ILC3s with hydrogen peroxide to induce oxidative stress, which is sufficient to activate XBP1, and this activation is suppressed by NAC, which is an antioxidant. And next, in order to look at what is the functional role of IL-1 XBP1 in ILC3s, I generate this conditional knockout mice by crossing IE1 alpha flux mice with rock-free mice. And at a steady state, I look at the numbers of the three subsets of ILC3s, including NKP46 positive, CCL positive, and double negative ILC3s. And here you can see in small bowel and the colon, the number of ILC3s was unchanged at steady state. However, when I stimulate the ILC3s with a combination of IL-23, IL-1 beta, here you can see the expression of both IL-22 and IL-17A were significantly reduced in the ILC3s from the knockout mice. And I also isolated ILC3s from colonic biopsies from healthy individuals came in for screening colonoscopy. And I treated those cells with IL-23 to induce the cytokines, and also the IL-1 inhibitor 4U8CO KIRA6. And here you can see the co-treatment with either IL-1 inhibitors significantly suppress the expression of IL-22 ILC3s. And in contrast, when I co-treated the human ILC3s with IXA4, which is the IL-1 activator, even under very low concentration of IL-23, this activator is sufficient to upregulate the expression of IL-22 in the cells. And those data suggest that the IL-1 XBP1 pathway functions as a positive regulator of cytokine production in both mouse and human ILC3s. And next, I use disease models to try to figure out what is the functional significance of this pathway. As we know that the C. diff infection is a common problem in patients with IBD. And here I use acute C. diff infection models on mice, which requires ILC3 functions by previous study. And basically, I give the mice and treatment of antibiotics and give them an infection, a garage of bacteria. And here you can see the knockout mice, the red squares, they had developed more severe disease as indicated by the weight loss, clinical scores, and shortening of the colon. And histology also give us a higher histology scores upon infection, as well as more goblet cell loss by action blue and PET staining. And upon infections, the ILC3 from the knockout mice produce less cytokine, including IL-22 and IL-17A, which is consistent with ex vivo stimulation I showed in the last slide. And the knockout mice also had reduced the expression of mucins as well as antimicrobial peptide, or RAX3G. The knockout mice had impaired recruitment of both neutrophils and inflammatory monocytes to the colon upon infection, which requires ILC3 derived cytokines such as IL-22. And the knockout mice had impaired epithelial barrier function, as shown by the facetextran permeabilization assays. And the knockout mice had increased translocation of the bacteria to the liver and mesenteric lymph nodes. And besides the infection models, I also challenged the mice with dextran sodium sulfate, or DSS, to induce acute colitis. And similarly, you can see the knockout mice had a more severe disease as shown by more weight loss, higher clinical scores, shortening colon with colitis, as well as higher clinical scores. And the knockout mice also had impaired epithelial cell proliferation, shown by the Ki-67 staining, as well as more epithelial cell death. And we also did bulk RNA sequencing of the clonic ILC3s with colitis. Here showed the knockout cells had impaired expression of ER chaperone, such as DNAJB9, as well as cytokines such as IL-17 and IL-22. And pathway analysis showed here there is a reduced cytokine cytoplasmic translation, as well as activation of lymphocyte and cytokine producing in knockout ILC3s. And besides acute phase of DSS colitis, I also look at how is the recovery of those mice after we discontinue DSS. And here you can see the knockout mice had a more impaired recovery after discontinuation, as shown by those red squares, both the weight loss and the clinical scores. However, when after I gave them an injection of IL-22, there's a significant improvement in terms of the weight in terms of weight change, as shown by those blue dots, suggesting that IL-22 plays an important role in this model in terms of the recovery from the disease. And I also look at the colon length as well as the clinical histology scores, which show the consistent results. And finally, besides the acute colitis models, I also look at how is this pathway affect ILC3s in chronic colitis models. So I cross the mice with RAC knockout mice and give them transfer of wild type T cells to induce chronic colitis. Here you can see the knockout mice develop more weight loss and clinical scores, suggesting a more severe chronic colitis upon T cell transfers. And the histology also showed there is more goblet cell loss as well as fibrosis, as shown by trichrome staining in the knockout mice. And given what we found in the animal models, so we asked, like, how can we apply those findings in human patients? And based on the findings, we hypothesized that the IL-1 alpha XBP1 pathway may indicate the body's responsiveness to IL-23 in IBD patients, and it sustains pre-barrier cytokines such as IL-22 induced by ILC3s under IL-23 suppression. So what we did here is we collected inflamed intestinal mucosa from patients with active Crohn's disease before they start using ustekinumab, which is a non-selective anti-IL-23 antibody, and we assess the response 40 weeks after initiation of the treatment. We performed flow cytometry to identify the mucosal ILC3s as well as the level of XBP1 in the ILC3s. Here you can see the patients with a higher expression of XBP1 in their ILC3s before the start of using ustekinumab actually had a better chance to respond to a therapy afterwards. So just to summarize what I just talked about in this project, we identified the non-clonic ER stress pathway that is mediated by mitochondrial loss, IL-1 alpha, and XBP1 in ILC3s during inflammation. And this pathway increases ILC3 cytokine production, which protects against infection and both acute and chronic colitis. And we think XBP1 positive ILC3s may become more useful biomarkers to predict the response to anti-IL-23 therapies in IBD patients in the future. And for the current studies, we kind of expanded our project to look at other immune cells that are activated by IL-23, including Th17 cells, Tc17 cells, gamma delta T cells, NKT cells, and mast cells. And based on the hypothesis that the phenotypes of those IL-23 responsive cells may control the IBD pathophysiology and potentially can predict the response to anti-IL-23 therapies. And I'm gonna switch gear a little bit and talk about related cell types in inflammatory bowel disease. So the group 2 innate lymphoid cells, or ILC2s, they are a different type of ILC that control the type 2 immunity, which are important for antiparasites, allergy, wound healing, and tissue fibrosis. And the prior studies show that ILC2 derived cytokines such as IL-13 and ARAC alleviate acute colitis in mice by improve their epithelial cell integrity. And the peripheral ILC2s increase in some IBD patients. And this cell type may also contribute to fibrosis of the liver or lungs. However, the role of the cell types in patients with fibrostenotic Crohn's disease or structuring Crohn's disease remain unknown. So I showed this figure before. The ER stress sensor IL-1 alpha is highly expressed in ILC2, although their function in these types is unknown. So in this particular study, I tried to answer 3 questions. First is, how is IL-1 alpha exhibition regulate ILC2 in mice and human? And how is the loss of IL-1 alpha and ILC2 impact colitis and fibrosis in mice? And is ILC2 related responses is linked to fibrostenotic Crohn's disease in patients? So to answer those questions, I generate these mice with a conditional knockout of IL-1 alpha in ILC2s. And first, I look at the cell numbers of ILC2 in the 3 parts of the GI tract: colon, small bowel, and esophagus. And here you can see, as a kind of, you can see the number of ILC2 actually increased in all the 3 parts of the GI tract in the knockout mice. And then I did a flow cytometry to detect the Ki67, which is a proliferation marker. And I found that the knockout mice actually express higher level of Ki67. This data suggests that the IL-1 XBP1 pathway actually suppress the proliferation of ILC2s in the GI tract. And next, I look at how those knockout mice produce cytokines upon stimulation in ex vivo. I sort ILC2s from the 3 different parts of the GI tract, and I stimulate them with a combination of IL-22 and IL-33, which are common cytokines we use to activate those cells. And here, by flow cytometry, you can see the ILC2s from the knockout mice express significantly higher IL-5, IL-13. Both of them are type 2 cytokines. And they also express higher level ARAC, which is an important growth factors. And I also collected esophageal biopsies from healthy individuals came in for EGD, and I purified the ILC2s from the esophageal biopsies, and I stimulated those cells with IL-33. And here you can see a co-treatment with IL-1 alpha inhibitor 4U8C is actually upregulated expression of IL-13. And in contrast, so the IL-1 activator IXA4 actually suppress the expression of IL-13 in human ILC3s. This data indicate that this IL-1 excipients pathway is a negative regulator of cytokine production in both human and mouse ILC2s. And next, I try to figure out what's the underlying mechanism of those changes. So I performed the bowel RNA sequencing of mouse small bowel ILC2 from the control and the conditional knockout mice. And here, one of the most suppressed gene came out from the sequencing is a gene encoding this retinoid X receptor gamma. And one of the most upregulated pathway in the knockout ILC3 is related to the fatty acid transport. And I look at the pre-literatures, and interestingly, before, people found that the retinoid X receptor gamma actually suppress the proliferation of ILC2s, while fatty acid metabolism is important for the activation and proliferation of ILC2s. And currently, I'm trying to establish what's the connection between those pathways and those two, and the retinoid receptor and fatty acid metabolism ILC2s. And for those condition knockout mice, I challenged them with acute DSS colitis to look at their response. And here you can see the knockout mice actually were partly protected from DSS colitis, acute DSS colitis, as indicated by the weight changes, clinical scores, and colon lengths. And here, the knockout mice also had more goblet cells at the end of DSS challenge. And the flow cytometry showed that the ILC2 from the knockout mice basically express higher level of IL-13 and ARAG. And this is consistent with prior findings that IL-13 and ARAG are protective against acute DSS colitis by induced goblet cell hyperplasia in the colon. And I also performed this T cell transfer induced colitis model to look at how those mice respond to chronic colitis and fibrosis. What I did is I transfer the mice with wild type T cell on day 0, and watch them for 40 days for weight changes, clinical scores. And here you can see the knockout mice actually developed more severe chronic colitis, as indicated by the more severe weight loss after the transfer of wild type T cells. And then in another group of experiment, I gave those mice an anti-IL-13 antibody. And based on the weight change, you can see the anti-IL-13 treatment actually is able to reverse the susceptibility to chronic colitis in those knockout mice. And I also look at the colon length as well as fibrosis by trichome staining, which showed the consistent results. And this study suggests that the overexpression of IL-13 by ILC2s in those mice actually is responsible for the worsening colitis and fibrosis in the knockout mice. And given the findings in animal models, we collected peripheral PBMCs from patients with active Crohn's disease with or without intestinal strictures. And what we did is we ran a flow cytometry to identify the ILC2s as well as the expression of IL-13 inside the cells. Here you can see the patients with stricturing Crohn's disease actually had a high expression of IL-13 in their ILC2s, which is consistent with the finding that IL-13 might promote the development of stricturing disease with chronic inflammation. And so for this project, for this study, we show that IL-1 XP1 suppress the proliferation and cytokine production of ILC2s. And the deficiency of this pathway in ILC2s protect against the acute colitis, likely by activating IL-13 and ARAG. And in contrast, this deficiency exacerbates chronic colitis and fibrosis, which is likely related to the upregulation of IL-13 in ILC2s. And we have some preliminary data to show that fibrostenotic Crohn's disease is associated with elevated IL-13 in peripheral ILC2s. And the current study is try to reveal what's the underlying mechanism of the phenotype in these animal models, and to determine what is the diagnostic potential of ILC2 related pathways in fibrostenotic Crohn's disease in patients. And yeah, and for the next part of my talk, I'm gonna switch gear a little bit. I'm gonna talk about this IBD associated complications called Crohn's-like disease of the pouch. And for this particular, for the data we have for this project, we are 100% focused on data from human patients. So yeah, as many of us know, for patient with ulcerative colitis who didn't respond to medical therapy or they develop malignancies, one of the common practice is to perform total colectomy with ileopouch anal anastomosis or IPA to create a pouch. And a lot of those patients, they develop like inflammatory complications after the surgery. And the most common one is called pouchitis. And there's another related but different inflammatory conditions called Crohn's-like disease of the pouch. And in most literatures, CDP is defined as de novo development of Crohn's-like phenotypes, such as pre-pouch ileitis, stricture or narrowing, and fistulization of the pouch body or extra pouch organs. And in the literature, people show that CDP can affect about 10% of the patients who had IPA for ulcerative colitis. The pathophysiology of this condition was unknown, and the diagnosis and treatment remain very challenging. So in this study, we try to answer 3 questions. First is, what is the cellular identity of Crohn's-like disease of the pouch? And how is CDP related to other IBD subtypes, such as UC, Crohn's, and pouchitis? And how are CDP-specific pathways may become new therapy for this condition? And for this study, we recruited 50 patients with a pouch, and we collected both, including patients with Crohn's disease of the pouch and normal pouch and ileum. And we collected biopsies from both pouch body and pre-pouch ileum. And we use those samples for different analysis, including single-cell analysis, cytoph or mass cytometry, immunohistochemistry, and ex vivo stimulations of cells isolated from those biopsies. And first, we use single-cell analysis to solve the different cell populations from the pouch body and pre-pouch ileum. And we identified 5 major cell populations, including T cells and innate lymphoid cells, B cells and plasma cells, myeloid cells, epithelial and stromal cells. And there are also a small population of mast cells and small populations of endocellular cells from both the pouch body and the pre-pouch ileum. And we identified that 14 out of 43 sub-cell populations, basically with altered frequencies in Crohn's disease of the pouch versus normal pouch and pre-pouch ileum. And also, we also confirmed those results using cytoph. And T cells actually is the largest cell populations from both the pouch and pre-pouch ileum. And we found several altered cell subpopulations in the CDP as compared to normal controls. Here you can see in the pouch body, there are upregulations of the regulatory T cells or Treg, increased Th17 cells, and the central memory T cells. And in the pre-pouch ileum, there are increased Treg, Th17 reg or regulatory, some people call it regulatory Th17 cells, and the follicular T cells. And we also performed the TCR sequencing, and we identified increased clonal expansion of T cells in the CDP tissues as compared to normal controls, and specifically in the population, including the Th17 prime CD4 T cells, Th17 cells, and KLRG1 CD8 T cells. And this data suggests this expansion, this shift towards effector T cells in the both CD4 and CD8 compartments in Crohn's disease, in Crohn's-like disease pouch samples as compared to normal controls. And we also look at the myeloid cell populations, which showed an increase of pro-inflammatory markers in the CDP samples, such as S100 and interleukin-1A genes. And we also identified this inflammatory monocyte population as shown indicated by the overexpression I1 beta, I1R, and CXCL2. And there is an expansion of inflammatory monocytes in both pouch body and pre-pouch ileum, as shown by the frequency of this population out of all cells, as well as the proportion out of the total myeloid cells. And we also show that the inflamed monocyte from the CDP tissues, especially the pouch body, express increased cytokine genes, including TNF, IL-1 beta, IL-23, which may contribute to the proliferation of Th1 and Th17 cells in CDP tissues. And similar to the myeloid cell population, we also identified an inflammatory monocyte population in the CDP tissues, which show increased frequencies in the CDP tissues compared to the normal controls. And the increased proliferation of inflammatory fibroblasts may contribute to the recruitment of myeloid cells and T cells to the tissue of CDPs and contribute to the chronic inflammation. And besides single cell analysis, we also collected paraffin-embedded blocks from independent cohort of patients with CDP and normal controls. And here we performed immunohistochemistry to validate our findings. And here we show that there is upregulation of both interferon IL-1 beta and CCL2 in the CDP tissues as compared to normal controls. And we also intend to elucidate what is the cell-cell interactions in CDPs by using this ligand receptor analysis. And among all those top interactions, we found there is a strong interaction between myeloid cell-produced cytokines such as IL-23 and IL-1 and the Th17 cells in the CDP tissues as compared to normal controls. And in order to validate those findings, we collected T cells from CDP and normal samples, and we stimulated those T cells ex vivo with IL-23 and IL-1 beta, and we detect the intracellular cytokines by flow cytometry. Here you can see the Th17 cells from CDP tissues produce significantly higher cytokines, including IL-17, TNF-alpha, and the interferon gamma, which may contribute to the chronic inflammation of CDP. And besides comparison between CDP and normal tissues, we're also curious what's the difference in the pathophysiology between CDP and pouchitis. So in this study, we performed a pseudo-time analysis by putting together the data of ours and also the single-cell data from this prior studies of pouchitis. And in the upper left panel here, we show the three monocyte populations from this study. And class 3, which is a classical monocyte population, and the cluster 4 and 5 both are inflammatory monocyte populations. And the pseudo-time analysis basically suggests the progression of class 3 to 4 and to 5 based on the expression of those some of the genes involved in monocyte activation, including CD44 and AF-kappa B1. And in the lower panel, you can see in the pouchitis tissues, most of the monocytes belong to class 3 and class 4. However, in the CDP tissue, there is a normal monocyte population, the cluster 5. And the analysis suggests that the cluster 5 might progress from cluster 3 and 4, which are present in the pouchitis. And this study is suggesting there is a continuum of monocyte development or activation from pouchitis and Crohn's disease of the pouch. And right now, we're trying to collect both tissues from those patients and to see if there's actual change from the different populations when patients first show clinical manifestations of pouchitis and later become more like a Crohn's CDP-like manifestations. And finally, we show there is an activation of the endoplasmic reticular stress actually in all major cell compartments of the CDP tissues. And you can see the upper panel shows a pathway analysis shows the ER stress and also unfolded protein response activation. And here, and in the lower panel, we show that those 6 genes actually were upregulated in all 5 major cell populations in CDP. And we also performed immunohistochemistry to verify the upregulation of both XBP1 and HX and BEP in the CDP tissues as compared to normal pouch and pre-pouch ileum. And finally, we performed another reanalysis of published database for Crohn's disease and ulcerative colitis. And we found those 2 databases, there is only ER stress in the epithelial cell, which is different from what we found in Crohn's-like disease of the pouch. So just to summarize what we found in this study, we found there's elevations in pro-inflammatory mediators and cells associated with Crohn's-like disease of the pouch, including IL-23, IL-6, the Th17 cells, and inflammatory fibroblasts. We also identified the complex cell-cell interactions between different cell types in CDP, which may contribute to the pathogenesis. We also found that ER stress induction in all major cell types in CDP, and they may potentially become new biomarkers for this condition. And for our current studies, we are planning to perform functional analysis of CDP-associated pathways using cell co-culture and mouse models. And we are also collecting pre- and post-IPAA samples, and we try to determine if any of those cellular responses and microbiome can predict the development of CDP in those patients. Finally, we are starting a clinical study to look at the potential of ER stress reducing agents. Here I put this chemical chaperone called tauroursodeoxycholic acid, or TUDCA, and we can see, and we will try to determine if this new drug can improve some of the disease in patients with refractory CDP to biologics. Finally, I would like to talk about some data we recently got related to another complications of IBD is called perianal fistulas in Crohn's disease. And perianal fistulas are abnormal tracts between the anal canal or rectum and connecting with other organs or the skin surface. And this is a very common complication which can affect about 1/3 of the patients with Crohn's disease. And the long-term remission rate is very low. And even with now many options of target therapies for IBD, still about 20% of those patients require prostatectomy due to their poorly controlled disease. And right now, we don't have a good disease-specific medical therapy for this condition. And the pathophysiology of this condition has been very poorly understood. So in this study, we hypothesized that the perianal fistula in Crohn's disease show a distinctive mucosal cell response as compared to idiopathic perianal fistulas or the cryptograndular perianal fistulas, as well as Crohn's disease without perianal disease. And we think that some of the unique pathways of PCD may become therapeutic targets in the future. So this is our study design. We recruited 3 groups of patients with perianal Crohn's disease without perianal disease, or patients with idiopathic perianal fistulas. And we collected biopsies from the fistula tract, rectum, and fistula opening for single-cell analysis, cytoph, and spatial transcriptomics. And we also performed integrated analysis or published IBD datasets. And due to time, I'm just gonna mention the single-cell findings from this study. So using single-cell analysis, we identify major cell populations from both PCD and IPF samples, including immune cell and stromal cells. And on the panel on the right, you can see most of the major cell populations were quite similar in frequencies between IPF and PCD, except there is a reduction of the mast cells in the PCD tissue, but we actually don't know what's the functional significance of this change. And in the myeloid cell population, the future clustering identified some detailed cell populations, and here you can see this upregulation of some pathogenic or pro-inflammatory genes such as STAT1, IFIT2, and NLRP3. And we performed the functional analysis, and on top of that, it's upregulation of interferon gamma responses in the PCD fistula as compared to the IPF. And other pathogenic pathway, including TNF-alpha, interferon alpha, and IL-6, JAK, and STAT3. And similarly, in the stromal cell populations, we identified 7 different subpopulations. And here you can see in the PCD tissue fistula tract, there's an enrichment of cluster 5, cluster 6, and cluster 7 as compared to the IPF samples. And those 3 cell population, stromal cell populations overexpress pro-apoptotic genes, including CD9 and ANXA1. And pathway analysis also show there's upregulation of interferon gamma responses, as well as the epithelial mesenchymal transition in the PCD fistula samples. And besides the fistula cells, we also want to look at how are the cell changes in the luminal surface of the GI tract. So we reanalyze this single cell data of rectal cells from a recent publication. And basically, we compared the rectal cells from patients with active or inactive perianal Crohn's disease. And similar to what we found in the fistula tract, the rectal cell from the active PCD also showed the activation of interferon gamma response as compared to the inactive PCD cells. And we did a further analysis of the rectal epithelial cells from this database. And here we identified 12 different epithelial cell populations, and 3 of them, including cluster 1, cluster 9, and cluster 12, are upregulated in the rectum from patients with active PCD as compared to inactive perianal disease or normal controls. And further gene expression analysis found that those 3 cell populations has increased the expression of genes that are related to interferon gamma response and TNF response. And we also look at the gene expression of genes related to the epithelial mesenchymal transition, and we calculate the EMT score. And here you can see those enriched epithelial cell population in active PCD has a higher EMT scores in the rectal epithelial cells. And besides the rectum, we also use a single cell data in this publication from last year. Basically, this study specifically look at the patient with ileal Crohn's disease. So we will reanalyze this data from ileal Crohn's disease, and we compared the expression of genes from patients with Crohn's disease with or without perianal disease. Here, interestingly, although we think ileum is quite far away from the anus, but those patients with perianal disease express higher level of interferon gamma response and epithelial mesenchymal transition, even in the epithelial cells as compared to those patients without perianal disease. And then since we found there is an increase in interferon gamma response in PCD, so we ask what is the source of interferon gamma in those patients. And in our single cell analysis, we identified the Th17 cell population in the fistula tract. And here we look at the gene expression of the Th17 cells from PCD and IPF samples. Here you can see the Th17 cells from PCD samples express a higher level interferon gamma as compared to the IPF samples. And the Th17 cells from the PCD fistula tract also express higher level of effector cytokines, including IL-17A, TNF, and CCL5, which may contribute to the chronic inflammation in those patients. And yeah, similar to what we did before from this study of ileal Crohn's disease, we look at how are the ileal IL-17 cells differ in those two patient population. Here we found that the Th17 cells in the ileum also express a higher level of interferon gamma in the patient with perianal disease as compared to those patients without perianal disease. And we also performed immunohistochemistry, try to validate some of the single cell findings. Here we stand for interferon gamma and phospho-STAT1, which is one of the downstream targets of interferon gamma. Here you can see the PCD fistula samples had a higher expression of both proteins by immunohistochemistry. Okay, just to summarize what I just showed for this part of our study, we found there the perianal Crohn's disease fistulas had an increased interferon gamma response in myeloid cells and fibroblasts. And this activation is likely driven by the increased interferon gamma positive Th17 cell in the fistula tract. And besides the fistula tract, we've also found that there is an increased interferon gamma response in the rectum and ileum of those patients with perianal Crohn's disease. And this suggests that there is certain luminal stimuli that drive the pathogenesis of perianal Crohn's disease in those patients. And we are also performing co-culture experiments between we're combining Th17 cells with epithelial organoids and fibroblasts isolated from those patients. And given the increased interferon gamma response in those patients, we are considering a study of anti-interferon gamma therapies in animal models of perianal Crohn's disease. And finally, I want to thank all the people who are involved in this study. So Kaiming Ma is a technician. He has been working with me for a year, and he basically contributed to a lot of data I showed today. And I want to thank my postdoc mentor, Dr. Marco Colonna, for his support. I've been a postdoc with him for 3 years. And also my mentors and collaborators, including Dr. Deepak, Dr. Choba, Davis, and Newberry in the GI division at WashU, and also at other institutes outside of WashU for their help. And I would like to thank my funding support from the Crohn and Colitis Foundation, the AGA, the DDRCC, the Doris Duke Foundation. The Pakula Foundation basically supported the single analysis of the pouch, as well as the internal support and the NIDDK KOA award. Yeah, thank you so much.
All right, thank you, Siyan. That was quite a amazing presentation. That's a lot of data and a lot of work. So congratulations on it. And congrats on your KO8 as well.
No, thank you.
All right. If anybody has any questions, they can send it to me, but I guess I can get started. I have a few questions, but I'm going to start with the latter half of your story. If you're finding in Crohn's disease of the pouch a single cell signature that is reminiscent of Crohn's disease, how is that not intuitive? Right? I would imagine that you would find signatures of Crohn's disease in what we term as Crohn's disease of the pouch, right? So
Yeah.
how do you plan on what is different about CDP compared to Crohn's disease, right? Like that's what is this data telling you? And I think along the same lines, for your perineal fistula, you're finding reactive activation in active disease versus inactive disease. And I would imagine that that's what you would see, right? So if you can sort of help me clarify, like, where do you think this research is headed, right? Because we're doing it to it. I'm trying to how do we separate things that we're finding that we would expect to find in a chronic inflammatory state
Mhm.
that looks like Crohn's, and it is Crohn's, that's why we call it Crohn's. How would you differentiate it from these other conditions?
Yeah, I think that's a great questions. For the first part, yeah, in terms of the difference between luminal Crohn's disease and CDP, yeah, that's one piece of data I didn't show. Like we basically perform, integrated analysis of our data and also the published luminal Crohn's data. And we actually look at the myeloid cell activation between luminal Crohn's and CDP, and we found they are actually very similar activation of those inflammatory markers between those two conditions. So I think from inflammatory myeloid cell standpoint, those conditions are very similar. They are much similar between luminal Crohn's and CDP if you compare CDP and pouchitis or UC. Another thing I found is very interesting is I think the last slide I mentioned the ER stress induction in CDP and other conditions. Yeah, as I show, like there's a ER stress induction in every major cell types or Crohn's disease over the pouch. But when we compare luminal Crohn's disease, we didn't see that activation. And especially, we didn't see a ER stress induction in immune cells or stromal cells of luminal Crohn's disease. So yeah, I think the short answer is actually I don't know how those two diseases are actually still related, but based on the data we have so far, I feel like the luminal Crohn's disease and Crohn's disease of the pouch looks like they are two different pathologies related to IBD. And I think one of the reasons is because of the big anatomy change, after the surgery. I think Crohn's disease of the pouch might have been driven by certain environmental stimuli, such as certain stimuli from bacteria that may contribute to the pathogenesis. But yeah, but definitely something we would like to see because right now we are collecting patient samples at the time of IPA and after that. And we're collecting both luminal mucosal samples and microbiome samples where we try to see if any of those host response or microbiome may potentially contribute to later development of CDP.
Yeah, I think that's going to
And
be critical, right? Like for you to be able to see this stress response in a uninvolved terminal ileal sample that is you're obtaining from a patient who has presumptively had UC, right? That would be the one that would really bring this together, right?
yeah, yeah. And for your second question, and you ask regarding the rectal samples, right? Yeah. So yeah, I agree with you. Yeah, because the original study basically they compared the I'm sorry, the rectal biopsies from inflamed rectum and non-inflamed rectum. Yeah, that's kind of probably not the perfect comparison. And I think because yeah, as you said, there is you are kind of compare like inflammation and without inflammation, you will definitely see some difference. So I think ideally, if we can basically compare those rectal mucosa either both with inflammation or both without inflammation, that will be more informative.
All right, thank you. Balfour, you have a question.
Yeah, first of all, congratulate you. That was quite a bit of information there. You mentioned the link between ileum and perianal fistulizing disease, and your last response was, talking about luminal bacterial. But I'm interested how you think those two are linked. They're very different ecosystems. And if you're thinking there's a microbial link, but without colonic inflammation, I was just wondering what you think those microbial stimuli might be that would be in those seemingly disparate areas.
Yeah, that's another great question. So, as what we found is like we found there is increased interferon gamma response in the ileal samples from patients without or without perianal disease. Yeah, this. Yeah, I have to say like I don't have enough data to answer the question, but if I have to guess. I think there might be a common trigger that actually present in the lumen of the GI tract. This could be, certain bacteria, which I don't know at this point, or some environmental triggers that kind of initiate the pathogenic signaling pathways from the lumen of the GI tract, which later leads to the development of fistula. So yeah, but as I said, yeah, right now, I don't know what actually could contribute to that. But I'm just thinking this could be a common kind of common pathogenic pathway that is not only present in the fistula tract itself, but also present in the lumen of the GI tract.
Yeah, it's just a little unusual that one might
Right.
skip the colon, that it wouldn't, in the perianal area,
That's right.
it
Yeah.
would be different. And you mentioned animal models
Right.
of fistulizing disease. I'm not really aware of that beyond the sample, and yet,
Oh,
single picture that I've seen for 20 years now of a hole in the perianal area.
yeah, that's an excellent question. Yeah, I was talking about Dr. Florence Celaro's group in Johns Hopkins. He recently developed a rat model of perianal Crohn's disease. Basically, what he did was he create some holes in the perianal area, kind of mechanically create some fistulas, and then they give the rats several cycles of TMBS to create a chronic inflammation, basically to keep the fistula tract open. I agree. Right now we don't really have a good model of perianal disease. I think the SEP1YT mice reported a small portion of those mice, like 5 to 10%, they develop perianal disease, but it's a very small portion of animals still. Yeah, and as you said, I think that's kind of like a big limitation and kind of limitation in the study of perianal disease at this point.
Yeah, I mean, it really has hindered the pathogenic studies. But anyway, congratulations. You not
Thank you.
only on your work, but you have a pretty impressive funding regimen here.
So in the interest of time, Sian, I'll ask one last question, I'll let you go. This is a little provocative. I've been hearing about IL-22 and ILC3s for over a decade now, and amazing papers. Where is this field headed? How is this going to impact us fixing Crohn's? And if you could sort of help me out a little bit as to where you think, how we're going to actually, how we're going to move. Thank you.
Yeah, I think that's a great question. So that's actually something I have been, brought up during our lab meetings. I will say this is, maybe I'm a little bit pessimistic, but I think I have to say there's some limitations there, because I think one thing about those ILC3s is it basically shares so many markers of T cell, especially Th17 cells. So I would say it's going to be very hard to specifically target this cell population. And there is, but this is something, some study which I found is very interesting, because they target the transcription factor called ROR gamma T, which is expressed in both ILC3 and Th17 cells. But there's some study shows the function and the activation dynamic is different between different cell types. So you might be able to inhibit that transcription factor during a certain stage of disease, but only targeting one, maybe the bad cell, but it still keep the good cell active. But the short answer is I don't know. anyway, we can specifically target ILC3 at this time, but maybe in the future we can see if something can distinguish ILC3s with other related T cells.
All right, thank you. I don't think we have any other questions. So thanks everybody for joining. Siyan, thank you again for your time.
Thank you.
And hopefully we'll see each other soon again. All right.
Awesome. Yeah.
Take care. Have a great evening.
Thank you.
Thank you.